Hello students, welcome to EPG Pathsala. I am Dr. Hari Mohan from Center for Medical Biotechnology, Maharishi Dayanand University, Rohtak, Haryana. Today we are going to discuss about a module, host pathogen interaction under paper animal cell biotechnology. The main objectives of this module is to understand what is host pathogen interaction. What is the significance of understanding host pathogen interaction, the resistance mechanism which the host exhibit against the pathogen, and the tools the pathogen have to invade in the host to establish infection, and finally what can be done in the host pathogen interaction in order to devise new drugs and vaccines for future use. In this world, we are surrounded by millions and millions of bacteria around us, and most of them are non-pathogenic. Microbes are present on our skin, microbes are present in our gut. We eat microbes, we inhale microbes, we swallow microbes, but then also most of these microbes will not cause disease. They are friendly bacteria with human being. Only a few pathogens cause disease or cause harm to the human being or animals. In order to elucidate how a specific pathogen is causing damage to the cells, to the tissues, or causing disease in the animals or human being, we need to understand exactly how the pathogen is interacting with the host and producing the factors which are damaging to the host. Host pathogen interaction is important in a way that it can open up the way of new strategies to prevent the pathogen from causing damage to host. So it have immense significance. It have immense potential for mankind. Host pathogen interaction will tell us how the pathogen is multiplying in the host. So what are the factors which are assisting the pathogen to multiply in host freely? How it is producing damage to host cells, whether it is giving certain kind of enzymes, whether it is producing certain kind of exotoxins, endotoxins, or what it is secreting that is damaging the host tissue. By means of host pathogen interaction, we can target the molecules or the epitopes specifically present on the surface of pathogen that can be used for vaccine designing in future. Host pathogen interaction is also indicative of what can be the most effective method to stop pathogen cycle in the body. So what is the most effective way, what is the most effective step, or what is the rate limiting step where we can stop growth of microbe or growth of pathogen without damaging the host cells. And lastly, host pathogen interaction will pave the way that how we can overcome the battle with pathogens. So how we can overcome the battle of pathogen resistance. So all these answers are lying in the host pathogen interaction. And lastly and recently, a very new concept of antibiotic resistance which have come in the way that how the pathogen is able to resist all the antibiotics in order to cause disease in the host. So this host pathogen interaction may solve the puzzle of antibiotic resistance also. So that's why it is very important to know exactly what is happening between host and pathogen. So this triangle shows how interaction of host with pathogen in a specific environment takes place. In the same environment, some hosts are more susceptible to a pathogen as compared to others, or the same host in different environment exhibit different susceptibility towards a pathogen. So whenever there is rapid change in environment, from summer to winter, heat to cold, or in rainy seasons, whenever environment changes, it puts stress on host, and that become a favorable condition for the pathogen to establish its infection in the host. So we can say that this host pathogen interaction is specifically determined by the environment, and disease triad, host pathogen and environment are three important pillars. These three things determine whether disease will happen or not. In addition to this, interaction of pathogens with other microbes present in neighborhood is also very important. So how the pathogen is interacting with its neighboring microbes, whether its neighboring microbes are giving space to pathogen for establishing infection, so that also determine whether the pathogen is able to cause disease in the host or not. About a century ago, Robert Koch identified diseases as a different stage from the healthy state, and he gave some postulates which are still followed about the causative agent of a particular disease. These postulates are known as cause postulates, and these are: the pathogen must be present in all cases of disease. So the microbe which is blamed for disease must be present in all the cases of disease. Second, the pathogen can be isolated from the diseased host and grown in pure culture. So from the tissue of diseased animal or human being, the pathogen can be isolated and can be cultured in pure culture by streaking or spreading. Third, when the pathogen from the pure culture is injected in the healthy organism, the same disease should appear. So third postulate state that the same disease should appear when pathogen from the pure culture is injected back in the healthy organism. And fourth one is, the pathogen must be re-isolated from the new host and shown to be same as the originally inoculated pathogen. So these four postulates, they still hold true in most of the cases. However, these are not true in some cases, such as mycobacterium infection, where the organism grow very slowly, or in some cases where the organism cannot be cultured in the media, or in case of viruses, the cause postulates are not true. So to refine them, molecular cause postulates have been proposed, and these molecular cause postulates are true in most of the diseased conditions. So when a microbe enter in the body, there are three possibilities. Either it can be in a situation of mutualism with the body, or it can exhibit commensalism, or it can show parasitism. So what are these different terms? First one, that is mutualism, is a condition where host as well as the microbe which is entering in the body, both are benefited. For example, microbes present in the gut of animals and human being. In that case, both microbe as well as the host is benefited from each other. Second type, that is commensalism, is a type of interaction between host and microbe where the host is not benefited, not harmed. So it is a neutral condition, but the microbe is benefited. So in this case only the microbe is benefited, there is no benefit or no loss to the host. For example, microbiota present in the eye. So there the microbes will get nutrition from the tear or from conjunctiva, but the host is not getting any benefit from that microbes. The third type is parasitism, and that is a dangerous situation where the pathogen is getting the benefit but the host is getting the damage. So the pathogen is getting benefit at the cost of host that is getting the harmful effect. For example, in case of malaria, where the plasmodium will cause disease in the host and it will multiply. So there is damage to the host but benefit to the plasmodium. So microbes are present all over the body and inside the body also. So they constitute an important part of our body. It has been estimated that our body contains 3 times more bacteria as compared to number of cells in our body. Recently, it has been found that microbes are determining our behavior in a way that the chemicals they secrete in small intestine or large intestine, and these chemicals are also responsible for change in our behavior. So microbiota is a term which is used to denote a group of bacteria present at a specific location in the body. So microbiota can be present in the small intestine, in the large intestine, in the eye, in the ear, and other body sites. Then also we can distinguish the microbiota into 2 types, indigenous and transient. Indigenous means that is normally present there. So it is normal microflora which is present at that particular location. So these are friendly bacteria and they prevent the pathogenic bacteria to establish their infection at that site. Whereas transient flora or transient microbiota include the group of microorganisms which occupy a site in the body for some time. For example, in case of diarrhea, when the normal microbiota is washed away, the transient microflora occupy that space. But as soon as the disease condition is treated, they are removed by indigenous microbiota from that site. So the site they occupy is termed as niche. So niche can be defined as a unique position occupied by a particular species perceived in terms of actual physical space occupied and function performed within the ecosystem. So what are the factors which affect growth of microbes in the body? First is nutrient availability. So microbes require nutrient for their growth. They require carbon source, nitrogen source, phosphorus source, sulfur source for their growth. So they require some of the nutrients in large quantity, that is known as major nutrient, and some nutrients in very small quantity that are known as micronutrients. So these micronutrients involves different vitamins or some kind of minerals such as copper, cobalt, etc. If sufficient nutrient is available, the microbes will grow at a higher rate. But if there is deficiency of nutrients or some inhibiting compounds are present, that will slow down or hamper the growth of microbes at that particular site. Second factor is physico-environmental parameters such as water activity, oxygen, pH, temperature. Water activity indicate the biological available water to the microbe. Oxygen, some microbes are aerobic, they require oxygen, where others are anaerobic, they don't require oxygen. So depending on the requirement of bacteria, whether they require oxygen or don't require oxygen, they will grow at the bodily site accordingly. pH, some bacteria love acidic pH, some require basic pH, where some grow very well at neutral pH. Similarly, most of the microbes, they are mesophilic. So most of the microbes which are pathogenic, they grow best at 37 degrees Celsius. But some bacteria require low temperature and some require more than 37 degrees Celsius for their optimum growth. Third factor is competition. When two or more than two pathogens infect the body at the same time, they will compete for resources, they will compete for food, they will compete for iron, they will compete for vitamins and minerals. So the bacteria which can uptake nutrients at a higher rate win in this competition and will first colonize the body sites. And fourth factor is host immune response. How quickly the host will put its immune response in function. So if the immune response is robust, the bacteria could not grow. If it is a weak immune response, bacteria could grow very easily at that body site. So how pathogen establish in the body, or how they establish their colony in the body. So first point is entry in the body. The pathogen can enter in the body via different routes such as mouth, nasal cavity, via contaminated food and water, via sexual activity, via injection, or via skin. So there are different routes through which the bacteria or the pathogen can enter in the body. After entry in the body, the pathogen will look for the suitable receptor and it will attach via ligands present on its surface to the receptor present in the body. So this adherence is most important when the pathogen enter in the body, and this adherence is mediated by certain factors which is important for its attachment and subsequent multiplication. So specific receptor on the body cells are required for that adherence. So adhesins are the site of attachment, that is specific receptor is there and to which the ligands present on surface of pathogen will bind. So this binding can be specific or it can be non-specific. Third step is invasion. After adherence, the pathogen will try to invade in the body throughout the cells or tissue of the host and will recognize specific factors. After invasion throughout the body, the microbes will start multiplying, and when the microbe is multiplying, it will secrete certain toxins, certain waste products which is dangerous for the body. So during course of multiplication, the microbe or the pathogens may secrete exotoxins, endotoxins or other enzymes which may damage the body of host. There are some factors known as virulence factors which are important for establishing the pathogen in the body. Important factors are adherence, invasion, degradative enzymes, exotoxins released by the bacteria, endotoxin released when bacteria die, induction of excess inflammation, evasion of phagocytic and immune clearance, byproducts of the growth of microbes such as gas and acid, superantigen released from the microbes, and resistance to antibiotic. So these virulence factor assist help the pathogen to grow well in the body and produce its effect. So first taking colonization factor. So these factors help in attachment of bacteria to the receptors on the cell. First one is capsule, which cause non-specific attachment. Second is surface receptors or target sites, which cause specific attachment. So there is specific binding of ligand with the receptor, and example of this kind of specific binding is fimbriae, formerly known as pili of Interbacteriaceae family. Chlamydia bind with the host cells via N-acetyl D-glucosamine, which is cell surface lectin. Protein adhesion of mycoplasma located in specialized tip structure, and hemagglutinin neuraminidase present in the influenza virus. So these ligands bind specifically with the host cells via a specific receptor. It indicates specific adhesion and receptor. For example, for group A Streptococcus, LTA-M protein complex is adhesin, whereas fibronectin act as receptor. In case of E. coli type 1 fimbriae, CFA1 fimbriae, P. fimbriae act as adhesin, whereas D-mannose, GM ganglioside act as receptor. In case of Treponema pallidum, P1, P2, P3, they act as adhesin molecule, and fibronectin act as receptor. In case of Vibrio cholerae, which cause cholera, type 4 pili act as adhesin, and fructose and mannose act as receptor. So pathogens have devised certain ways to damage host. There are three main types of molecules which are released by pathogen, and these are degradative enzymes, endotoxin and exotoxin. These degradative enzymes are certain kind of protease such as hyaluronidase, cellulase, etc., which cause degradation of proteins or intercellular matrix. So breakdown of intercellular matrix result in faster spread of microbes across the body. In addition, they will destroy the cell also. They will kill the cells also and produce the damage. Second type, that is endotoxin. Endotoxin, as the name indicate, they are present inside the bacteria itself. Actually, endotoxin are component of bacterial cell wall. So they are not released when bacteria is live, but only released when bacteria die. So after death of bacteria, its cell wall components are degraded and released in the blood of host. So from the cell wall component, it is lipid A which act as endotoxin, and it produces many effects in the host. So these effects are non-specific, such as fever, soak, and edema. So many things happen in response to release of endotoxin. The third one is exotoxin. Exotoxin are released when bacteria is rapidly multiplying and secreting the toxin outside the cell. So these exotoxins, they also cause damage to the host. The mechanism is different in different conditions, but these exotoxins, they are responsible for the disease a particular bacteria cause. So endotoxins, which are released after death of microbe, have n number of effects on the body. When endotoxins are released in the body, they increases the temperature of body, and that result in fever. These endotoxins also result in leukopenia or leukocytosis depending on the concentration of endotoxins. They also have some metabolic effects on body, such as hypoglycemia and hypotension. They result in death of cells in the immediate vicinity, and they can even lead to septic shock. So when endotoxins are released in excess concentration, they can lead to septic shock. They can even lead to disseminated intravascular coagulation or DIC. Excess concentration of endotoxin in blood may result in activation of blood coagulation pathways, and activation of blood clotting pathway result in clotting of blood in the vessel itself. So that may result in blockage of vessel and infarction of the tissue to which the vessel is supplying. It can also result in necrosis of organs. Where the endotoxin has released, it can even lead to death of certain portion of the organ. Certain microbes secrete exotoxins. So exotoxins are released when bacteria or the pathogen is multiplying. Generally, exotoxins have two subunits, one large subunit and one small subunit. This large subunit will bind to the target cell and it will cause moving the small subunit inside the cell. The small subunit will exhibit its toxic effect. For example, tetanospasmin, tetanolysin, cholera toxin, LT toxin, diphtheria toxin, botulinum toxin. So most of these toxins have two subunits, and that's why these are also known as AB type of exotoxins. So these exotoxins are the cause of tetanus, cholera, diphtheria, botulinum disease. So how we can differentiate endotoxins from exotoxins? As the name indicates, exo means outside, endo means inside. So endotoxins are integral part of cell wall and they are only released after death of bacteria, whereas exotoxins are released from the bacterial cell when it is multiplying. Endotoxin is lipopolysaccharide in nature. So it is lipid A which act as endotoxin, whereas most of the exotoxins are protein in nature. Endotoxins are heat stable. They can tolerate temperature up to 100 degrees Celsius, whereas exotoxins are heat labile. They cannot tolerate temperature above 50 degrees Celsius. The antigenicity of endotoxin is questionable, so sufficient immune response is not generated against endotoxins, whereas exotoxins exhibit antigenic and immunogenic properties. That's why toxoids cannot be produced from endotoxin, but toxoids can be produced from exotoxin. What is toxoid? Toxoid can be defined as inactivated toxin that can be used for vaccination. So endotoxins have many effects on the host, where exotoxin have specific effect on the host. Endotoxins are only produced by gram-negative organisms, whereas exotoxins are produced by gram-positive as well as gram-negative organisms. So apart from direct damage, pathogens also cause indirect damage to the host. So in indirect damage, pathogen is not responsible directly. For example, in case of hypersensitivity. In case of hypersensitivity, bacteria or the pathogen is not in the scene, but then also it may be responsible for the occurrence of hypersensitivity in the host. Hypersensitivity is basically of four types, that is 1, 2, 3, 4. Hypersensitivity 1 is mediated by IgE. It is also known as immediate type of hypersensitivity. Type 2 hypersensitivity is also known as cytotoxic reaction, where specific destruction of cell by autoantibodies occur. So it can also be termed as autoimmunity for certain kind of cells and tissues. Type 3 hypersensitivity is mediated by immune complexes. When the number of antigen and antibody combining is very high, so that high concentration of antigen antibody complexes, they get deposited in different organs such as kidney. So their deposition of this antigen antibody complex will activate complement system, and that will damage the cells which are in neighborhood of that antigen antibody complex. Lastly, type 4 hypersensitivity is cell mediated, and it is delayed type hypersensitivity which is cell mediated, and it result in granulomatous inflammation. So the tissue destruction occur and a granulomatous tissue is formed at the site of infection. So these hypersensitivity are example of indirect damage produced by the pathogen. So the pathogen has to win with the host defense, has to overcome host defense. Only then it can survive inside the host and can cause disease. For this, pathogen adopt many strategies such as antigenic mimicry, antigenic masking. So the bacteria will coat itself with certain kind of matrix, certain kind of slime layer, and this coating mimic that it is a part of body itself. For example, Streptococcus pneumoniae, Neisseria meningitis, Haemophilus influenzae, they have polysaccharide capsule around them, where Bacillus anthracis have polypeptide capsule. So it is a mode of masking its antigen or doing mimicry of its antigen. Second method is evasion or incapacitation or avoiding the phagocytosis. So the pathogens may secrete certain kind of phagocytosis inhibitors, and these phagocytosis inhibitors decrease or inhibit the phagocytosis by the killer cells. So when there is no phagocytosis, the pathogen can survive very easily inside the host and can multiply. Hosts also exhibit resistance to microbes. It is very surprising that we are surrounded by thousands and thousands of microbes everywhere. But then also there are few microbes which can cause disease in human being and animals. That is due to the robust immune response animal or human being have. So hosts also resist colonization by microbes. Hosts have many strategies to prevent the pathogen to enter in the body. The first one is external. The physical barriers, they prevent the pathogen to enter in the body, such as skin, respiratory tract, gastrointestinal tract, genitourinary tract. So these covering prevent the entry of pathogen inside the body. Sweat gland, fatty acids, pH, indigenous competitive flora, peristalsis movement, hair, cilia, urinary flushing, mucus. So there are different mechanisms how the colonization of pathogens are prevented in the body, and they are flushed out of the body. Certain kind of active elements such as lysozyme in saliva, spermine in semen, and IgA in mucosal secretory antibody. So they prevent localization of pathogen in the body and thus protect the body from pathogen infection. If the pathogen is able to enter in the body in spite of external resistance by the host, it will try to attach to the receptor present on host cell and will try to invade and multiply. But here also the host body have robust resistance mechanism against the pathogens. The mononuclear cells, monocytes and macrophages, and polymorphonuclear cells, also known as PMNs, they are important phagocytic cells. So they will target whatever pathogen will enter in the body, and they will engulf the pathogen and will destroy them. So this system also known as reticulo endothelial system, is a robust mechanism of phagocytosis of whatever is coming from outside, whether it's pathogen, whether it's a virus, whether it's bacteria. Everything is destroyed by the body to prevent the disease to happen. Apart from external and internal resistance mechanism, hosts have certain other mechanisms also, such as inflammatory response. So whenever pathogen will enter in the body will exhibit inflammation against the pathogen. Inflammation involves increase in temperature of body and localization of pathogen. So the cells in the immediate vicinity of pathogen will start exhibiting inflammation, and that inflammation will prevent spread of pathogen from that particular site. Increase in the temperature also result in increased activity against the pathogen. Second, phagocytosis that we have discussed. Third, cytokines. In response to pathogen infection in body, a number of cytokines are secreted, and these cytokines, they stimulate various kind of cells in the body and prepare the body for the upcoming danger. So the body prepare itself for war against pathogen. Another very important component is complement system. Complement system in the body prevent the pathogen to invade and to multiply, as complement system in conjugation with the antibodies present in the body cause puncturing in the bacteria. It form pores and thus kill the bacteria. And the last is antibody production. So our B cells, they get converted into plasma cells, and these plasma cells secrete specific antibodies. These specific antibodies bind to the epitopes on the surface of pathogen and thus prevent its growth and kill it. So we can say host also exhibit a high amount of resistance for pathogens. So for pathogens, it is not easy to establish its infection in the host in spite of all their virulence factor. So it is all war between host and pathogen, how they interact, what is the environmental condition, and how much is the host under stress. All these factors determine what will be the outcome of host pathogen interaction, and whether the host is able to prevent the pathogen to enter in the body, or whether the pathogen is able to win over host and cause disease in the host. So we can summarize the module as the microbes infect many organisms, many animals, many human beings, but only few get the disease. You will be surprised to know that when Mycobacterium tuberculosis infect 100 patients, the disease appear only in 10 patients actually. 80 to 90% of patients don't exhibit sign and symptom of tuberculosis. It's very interesting, means how the bacteria is interacting with the host and how the host is taking care of bacteria. So in 90% of the cases, there is no sign and symptoms. Sign and symptoms appear only in 10% of cases. So this is actually interaction of host with the pathogen in a specific environment. So this host pathogen environment triad of disease is important to be considered. Till date, host pathogen interaction for all the microbes has not been elucidated because it is a very complex process. And if we are able to solve it, if we are able to elucidate this complex puzzle, it may be possible that we may get a totally new set of drugs. We may change the way how the diseases are treated currently, and we may have the treatment strategies and prevention strategies for the disease which are considered as incurable nowadays. So we can say that the host pathogen interaction is very important to understand, and this in-depth understanding of host pathogen interaction will open the door of immense possibilities, and that will make the world free of disease and all will be hale and healthy. Thank you.